Good morning everyone. So today topic is nucleus, also known as brain of the cell, or also we can use another term, master of the cell, because this nucleus which coordinate and which regulate and which control almost all cellular activity in the cell. Hence nucleus is known as a brain of the cell or master of the cell. And this nucleus also known as karyon. The study of nucleus is called karyology. Study of nucleus is called karyology. Here important note, nuclear division. So in mitosis and meiosis there is a division of nucleus and there is a division of cytoplasm. If first nuclear division occur, then it followed by the cytoplasm division. And here nuclear division is called cyto, but the study of nucleus is called karyology. Here now we move on discovery of nucleus. Discovery. So first discovery of nucleus made by Robert Brown in orchid plant. In orchid plants, root cell. Orchid plant root cells in 1831. And one more scientist here will need to discuss about Hammer. He discovered nucleus in Acetabularia. This Acetabularia is the largest unicellular algae. It is the largest algae. So Hammerling discovered nucleus in Acetabularia algae, and Acetabularia is the largest unicellular algae. And not only he discovered nucleus in Acetabularia only, he proposed karyo index. This karyoplasmic index is also known as nucleo ratio, or simply we can write like this: N ratio, or Ki means karyoplasmic index. And why he's proposed this karyoplasmic index in nucleus in Acetabularia means, generally this karyoplasmic index which describe the relationship between the volume of nucleus and the volume of cytoplasm and the volume of cyto in a living cell. Here this karyoplasmic index which mainly describe the relationship between volume of nucleus and volume of cytoplasm in a living cell. Why this volume of nucleus and volume of cytoplasm in living cells to know be? Because this karyoplasmic index which it is a vital indicator. It's a vital indicator of cell health, how cell become healthy, cell health, and also it is a vital indicator for cell maturity, and very important also it is an indicator of phases of cell cycle. In phases of cell cycle we have different phases, we will discuss in another video lecture. So to be know the cell conditions like cell health, cell maturity, and phases of cell, there is a must and should be based on this karyoplasmic index only. And there we have one formula for karyoplasmic index. Here the formula is Ki is equal to karyoplasmic index Vn by Vc minus Vn. So this is the formula for karyoplasmic index. So where here Vn indicate volume of nucleus, and here Vc indicate volume of cyto. And very important note here I am writing, very important note, if so here note, if low Ki indicates nucleus size is small and meta cell become stable. If high Ki, high karyoplasmic index, which indicate the nucleus become large and cell become metabolically unstable. Cell become metabolically unstable. So likewise, if the index of low and high, which indicate the different aspects, like low Ki indicate the nucleus become size and metabolically stable, and high Ki indicate the nucleus become large and cell become metabolically unstable. So this is all about the karyoplasmic index, how, why they are, and how they are influence the cell health, cell maturity, and phases of cell cycle. So this is all about the Hammerling only. And one more scientist here we need to remember in nucleus, so that is Walter Fleming. Here I'm writing, Walter Fleming. He used basic dye, like he used aceto or aniline. These are basic dye. Why? They stain to nucleus. Then he given the term for nucleus as a chromatin. Very important. The chromatin name or term was given to the nucleus by Walter Fleming by using a basic dye called acetocarbamide and aniline to stain the nucleus. Chromatin. Generally, this chromatin, it can be visualized only during interphase. Remember, note here. Basically, this chromatin can be visualized during interphase only. Whereas later we know that the chromatin later condense together form chromosome. Whereas chromosome can be visualized during dividing phase. So here dividing phase means mitosis or meiosis. So remember, the chromatin can be visualized only during inter. So likewise, the Walter Fleming, he used the basic dye to stain the nucleus and he given the term chromatin. Yes. So all these are the discovery of various scientists to the nucleus. Now we will move on very important types of nucleus. Here I am writing types of nucleus. So based on presence or absence of nucleus in cell. Based on presence or absence of nucleus in cell. Generally, there are only few cells that do not have any nucleus. Such nucleus absent cells are called enucleated. Here I am writing enucleated and nucleated cells. Enucleated means absence of nucleus. Absent of this. So here nucleated means presence of this. So generally, enucleated cells are like mature RBC cell, erythrocyte cells, and one more sieve tube cells. So generally, the sieve tube cells is a phloem cells of vascular bundles, and one more important cells. It also doesn't have any nucleus. That is most important. Prokaryotic cells include bacteria. So these are the important enucleated cells. And now coming to nucleated cells. Generally, nucleated means the cells which contain nucleus, and here there are different types of cells contain different number of nucleus, based on which we can classify them into different categories. For example, if a cell has a single nucleus, then it is called uninucleated cells. Uni, and it has both two, then called binucleated. If a cell has more than two nucleus, such cells are called multi. So here the important of uninucleated cells, like mostly plants and animals. So plants and animals contain only single nucleus cells. Generally in plant, the nucleus is present periphery, but in animal, the nucleus present at the center. Very important, center. Now coming to binucleated nucleus. Generally these binucleated nucleus are present in fungi only in two classes, like Ascomycetes fungi and Basidiomycetes fungi. And not only fungi, it also present in a protozoa organisms, that is Paramecium. In Paramecium also we have seen two nucleus. It is a protozoa organism. In Paramecium again there are two nucleus are there. One is small size nucleus called micronucleus, micro, and large size called macro. Generally micronucleus helps for reproductive structure. It is helps in reproductive nature. And macronucleus is for vegetative cells, vegetative nucleus. Important thing here, in Paramecium we have two nucleus, hence they are binucleated conditions, in which macronucleus and micronucleus small in size, they are only for reproductive nature, means they are 2n. Reproductive nature always 2n, and they are vegetative like n. Okay, right. Likewise, in binucleated I have seen fungi like Ascomycetes, Basidiomycetes, and also in protozoa organism, that is Paramecium. Now coming to multinucleated conditions. Generally these multinucleated conditions are seen. Here I am writing here multinucleated, multi cells. These multinucleated cells are generally present, most important here, skeletal muscles. Skeletal muscles are also called striated muscles or voluntary muscles, and also which is present in coconut endosperm. And also multinucleated conditions are present in, very important, tapetum is the innermost layer of anther wall or microsporangium. So likewise in skeletal muscle, in coconut, endosperm, and tapetum, in which we have seen, in these cells we have seen multinucleated condition. This multinucleated condition also called xenocytic condition. Xenocytic, here I am writing xeno conditions. So the sinusitic or multinucleate conditions are seen in this organism, especially skeletal muscle, coconut muscle, and tapetum is the innermost layer of microsporangium or anther wall. So this is all about the different types of nucleus based on their presence or absence of nucleus in the cells. Now again we move on another type of nucleus, and this nucleus which is mainly based on nuclear membrane. Yeah, so types of nucleus based on nuclear membrane. Yeah. So here first pronucleus and eukaryotic nucleus. Here prokaryotic nucleus are also called nucleoid, and eukaryotic nucleus they are also, we can use the term true nucleus. So let us discuss the major differences between prokaryotic nucleus and eukaryotic nucleus. And here in prokaryotic nucleus, nuclear membrane is absent. Very important. Nuclear membrane is absent around the genetic material. How means like this? For example, here it is a best example for prokaryotic is bacteria generally. So bacteria are prokaryotic organism. Generally it has their genetic material like this. The main gene chromosomes are genetic material, and generally each and every genetic materials are covered with a membrane. Such membranes are called only nuclear membrane. So around this genetic material there is no membrane are found. Hence nuclear membrane is absent around this genetic material, and such nucleus are called nucleoid or prokaryotic nucleus. And not only these differences, and one more important character it doesn't have, that is every genetic, generally I'm talking about generally, every genetic material must and should contain DNA, and every genetic material always DNA, and this DNA must and should contain histone protein, generally histone protein. But in case of prokaryotic nucleus, and here the genetic material that is DNA without histone protein. So genetic material that is DNA without histone protein, such type of nucleus also we can said to be as a nucleoid or prokaryotic nucleus. And generally this type of prokaryotic nucleus are seen in prokaryotic organisms. Proorganisms. So here prokaryotic organisms generally include like Monera kingdoms. Monera kingdom. In Monera we have different types of classes. We will discuss in another video lecture. In Monera, best example Monera kingdom organism is bacteria. And now coming to eukaryotic or true nucleus. Here, yeah, in eukaryotic, very important. For example, it is a cell, and every cell has genetic material in the form of DNA. So this is the genetic material, that is DNA. And these DNA are genetic material which is surrounded by a nuclear membrane. Yeah, like this, surrounded by a double membrane nuclear membrane. See, like this. See, this is what nuclear membrane. So this nuclear membrane present around this genetic material or DNA. Such type of nucleus are called eukaryotic nucleus. So here I am writing here nuclear membrane is present around genetic material, that is DNA. And one more differences here, every genetic material, that is DNA with histone protein. Very important, with histone protein means their genetic material with made up with DNA and histone protein. But here without histone protein. Remember, here in prokaryotic without histone protein, but here with histone protein. Here nuclear membrane is absent, and here nuclear membrane is present. So that is the major differences between prokaryotic nucleus and eukaryotic nucleus, or prokaryotic organism or eukaryotic organisms. Hence, due to presence of nuclear membrane and due to presence of genetic material with histone protein, such type of nucleus is called true nucleus. Generally this type of nucleus are generally mostly found in higher organisms, so like algae and each and every plants group, like plants, and even though in fungi also, and animals. So all these organisms, they are contain eukaryotic nucleus. Okay, so likewise we have types of nucleus based on presence or absence of nuclear membrane, and also genetic material with or without histone protein. With or without histone protein, we classify them into two categories. Okay, right. So yes, and now we move on very important concept, that is structure of nucleus. Yeah, here structure of nucleus. Yes, so in this structure of nucleus, generally the nucleus mainly contain two important parts here. So that is nuclear membrane and nucleoplasm. how means, for example, see, I have draw a cell, and every cell has a nucleus like this. Okay, so this is a nucleus, and this nucleus is a double membrane structure. Here I am drawing one more, yes, double membrane structures. And see, these two are nuclear membrane, and inside the nuclear membrane, this empty space which is filled with a plasma means fluid, and this structure is called nucleo. Likewise, every nucleus has important two structures. They are nuclear membrane and nucleo. Now, generally we have two nuclear membrane. What they are? Outer nuclear membrane and inner nuclear membrane. So this is the outer nuclear membrane, and this one inner nuclear membrane. And between these two nuclear membrane, there is a space here. This space here, I'll show you this space, and this space is called perinuclear space. Sorry, perinuclear space. Okay? Perinuclear space. So this perinuclear space present between two nuclear membrane, outer and inner. So generally the space 10 to 50 nanometer in size. 10 to 50 nanometer in size. And these two membrane, outer and inner two membrane, they are fused to form. They are fused together and form a pores, means holes. Like there are different holes or pores are present, and these pores are formed due to fuse by two membrane, and these pores are called nuclear pore. Here, fuse of these two nuclear membrane to form nuclear pore. And why these pores are formed means they helps, these nuclear pores helps to passage for the bidirectional, here by movement of bidirectional, here I am writing bidirectional movement of RNA and protein between nucleus and between cytoplasm. Here, for example, inside the nucleus there is a formation of RNA. After this RNA, after formation of RNA by transcription process in the nucleus, it must and should goes back to the cytoplasm. Here, this region is called cytoplasm. This region is cyto. Why means for translation process, it must and should enter to the cytoplasm RNA. So this RNA will goes out from nucleus to cytoplasm through this nuclear pore, and this RNA in cytoplasm, then it undergoes translation process to synthesis protein. So once the protein will synthesis, then again this protein will enter into this nucleo. So likewise, there is a transport of or movement of molecules between nucleus and cytoplasm. There must and should be present nuclear pore. So hence this nuclear pore allow bidirectional movement. Okay, so this is all about the nuclear membrane. And now coming to nucleoplasm. Generally, this nucleoplasm contains two important structures. They are inside this nucleoplasm here. Here inside the nucleoplasm, we have some small spherical-like structures like this, small spherical-like structures, and they are called nucleolus. And also, in which we have a elaborate network-like structure like this, elaborate network-like structures, and this structure is called chromatin. Very important. So there are two important structures are present inside the nucleoplasm. One is spherical-like structures called nucleolus, and one more important structure, that is, it is a elaborate and network-like structures which is present inside the nucleoplasm, that is chromatin. So now we discuss some important about nucleolus. Generally, nucleolus is an active site for ribosomal RNA synthesis. Very important here, very important. So what is the importance of this nucleolus means? It is a active site for ribosomal R synthesis, means rRNA synthesis. So rRNA synthesis taking place only because of this nucleus. Hence, this nucleolus is also known as ribosomal factory. So which cell organelles is known to be as a ribosomal factory means nucleolus, because of it is an active site for synthesis of ribosomal RNA. And remember, this nucleolus is a membrane bound cell organelle, means it doesn't have any membranes around it. So a membrane and cell organelles. Okay, so this is the two important, and it is a spherical in shapes. Remember, this nucleolus is a spherical in shapes. Spherical means round shapes. Okay, spherical in shape, and it is a membranous bound cell organelles, and it is active site for ribosomal RNA synthesis, that is ribosome. Hence, this nucleolus is called ribosomal factory. Now we move on chromatin. Yes, generally about chromatin. Yeah. Yes. Okay. So this chromatin. about chromatin during interface only this chromatin can be visualized, very important. And how they are visualized means this chromatin which is like loose like this. For example, here again we will draw, think it is a nucleus only, and in which we have chromatin like this chromatin. And how this chromatin will be visualized means during interface, very important. During this chromatin can be visualized only during interphase, but chromosome visualized in dividing phase or mitosis and meiosis. And here how they are visualized means they are loose elaborate network of nucleofiber. They are nucleoproteinous fibers means they are, see how this appear means they are loosely and they are elaborate network like structures, and they are made up of with nucleofibers. Here nucleoproteinous fibers means generally these chromatins are made up with two important structures, like they are nucleic acids and also proteins. Like virus also it is a nucleoproteinous structures, they also made up of with nucleic acid and protein. And also ribosomal RNA or ribosomes also it is a nucleoproteinous structures. How means they are also made up of with nucleic acids and proteins. So likewise chromatin also nucleoproteinous structures means it is made up of with nucleic acids and protein both. So here the nucleic acid, both the main nucleic acid and this nucleic acid act as a genetic material, that is DNA. So DNA is the main genome or genetic material of this chromatin. And also one more nucleic acid is present, and but it is act as a non-genetic material, that is RNA. Both DNA or RNA is present, but in virus either of the nucleic acids are present, either DNA or RNA. But in chromatin, both DNA and RNA nucleic acids are present, but in which DNA act as a genetic material and RNA act as a non-genetic material. Very important. Non-material, but DNA act as a genetic material. And both nucleic acids are present in chromatin. And coming to here protein. So here we have two types of protein. One is histone protein, also histone protein, and also non-histone protein. Very important. Non-histone proteins, histone and non-histone protein both are present. So likewise nucleic acids and protein, in nucleic acid DNA and RNA, in protein histone and non-histone, all these four important chemical composition of this chromatin. Okay, so likewise there is a chromatin which is present in the form of network of nucleoproteinous fibers inside the nucleo. So that's it. So with this we have complete entire concept of nucleus. So here I have provided three important MCQs here. Okay. So examine the following statement regarding the historical discovery and observation of the nucleus. So here statement, there are three statements are there. So which of the following option is correct? Choose the correct options. And one more MCQs. Yeah. So here consider the following statement regarding the structural component. Just now we discussed structure of the nucleus. Here structural component of the nucleus. So here also there are three statements. Which of the following options represent the correct set of statement? One more MCQs. Yeah. Regarding interface chromatin, just now we discuss here interface chromatin. Generally chromatin visualize or appear only in during interface. So which of the following statement are correct? So read this three statement 1, 2, 3 and select the correct option. So with this we have complete and the entire concept of nucleus, and with related to three MCQs. They are highly important NCERT based MCQs, multi-statement MCQs, and that's it. So if you like my videos, please subscribe my channel and share this video to your friends. So thank you. Have a nice day.